All right. Good morning, everyone. Welcome to day 3 of the CPATH Global Impact Conference. We're so grateful that you guys could be here again today. We'll be talking about navigating novel therapies with novel evidence sources. So right now I'd like to welcome Klaus Romero up to the stage to lead us off with some opening remarks. I think we've done all the housekeeping. Just a reminder to silence any devices. But other than that, let's go ahead and get started.
Okay. Sure, I turn this on. Okay, so yeah, we're going to start with this moment of remembrance because of the dates of today. I think everybody remembers where we were that day. I was on a shift in the ICU as a very idiotic resident, and thought everybody was joking when they were talking about this. And I was dealing with a really complicated case. It was only after I came out of dealing with a really complicated airway management situation that I saw what had happened, and everything had went down already. But let's give the victims and the families of this tragic day a moment of silence. Okay, so last day of the conference. We're going to talk about novel solutions for modern drug development. But modern drug development with novel solutions means you saw day 1 all about going early into the disease process. Day 2, connect the dots for tell me the context of use. Let's make sure that we understand what questions we're going to answer. But it all brings us to today, because if we're going to go early and we're going to pay attention to truly having a positive impact in modifying diseases, we need to pay attention to the kids. So we're going to be talking about lots of rare diseases and lots of pediatrics today. And that's part of the reason why I broke protocol, and I had a different yellow tie in store for today. But given the inspiration that I got from several of our advocates of opening up their lives, I'm going to open up my life a little bit. So this is 1995. The hair is gone, the youth is gone, and some of the energy is gone. But that's me on stage at a festival where we opened for Metallica. So that's part of my life. And so somebody yesterday made this comment to me, and I'm gonna steal it, and that's going to be my self-description. I'm a happy musician, happens to be a struggling clinical scientist. So some of you may have seen me in Zoom calls and Teams meetings with a background that looks rather professional in my home office. But I'm going to show you what you don't get to see that is on either side of my computer setup on my cameras. This is one side. And 2 of those guitars I built from scratch, but that's what keeps me sane. And this is the other side. So that's why I'm a happy musician, happens to be a struggling clinical scientist. But it's thanks to you that I wake up every day and I put the effort to transform drug development and modernize things. And Kristen, this goes to you. I just opened up my life thanks to your lesson from yesterday. Okay, but this is the fantastic human being that signed the zebra guitar and has a rare disease, inclusion body myositis. He was very inspirational last night, not just because he played his heart out, but because he sent a really compelling message about seizing life and helping others. So that's why I'm wearing the T-shirt with pride. And as I said, a diagnosis does not define you, and certainly does not define the children that suffer and the children that we can help. Especially because the children and everybody else that is in need has an army behind you. And so this is the group that took me up on the offer of seizing the tickets and coming to the concert line last night. We had a blast, and we're going to have a blast today connecting further dots for the sake of children and modern drug development. So with that, I want to stress a couple of facts. This is your effort. The public-private partnership model that we coordinate is for you. You are part of the 1,600 scientists that collaborate with us every single day. You are a part of CPATH as much as any employee of CPATH. And that's what drives us to do what we do and help the people in need. And we will continue to partner to de-risk and accelerate drug development, be the nonprofit that can add value to industry, to the patient advocates, to the clinicians, the academic scientists, and the regulatory agencies. So that's why we have the mission we have, and that's why we're here today. So thank you for being here. Thank you for participating. As I said, this is your thing. You're as part of this as any one of us at CPATH. Okay, so with that, let's move on to our panel conversation. So if I can invite our panelists to join me on stage. We're going to start the day with a pretty cool dialogue on evidence and how to communicate that evidence in a meaningful way. All right, so by the way, Peter Marks is stuck in traffic, but he gave us the green light to get going. Oh, and there he is, on the nick of time. Perfect. Okay, so while we get started, so we have a pretty awesome representation of multiple sides of the equation at the FDA. How many times have you heard the phrase, "Oh, FDA said," fill in the blank? What does that mean? In what context? Which group? To answer what question? So you're going to see that represented across the panel today from multiple parts of the agency. And then we have our colleague that has seen both sides of the ocean in action and somebody that really cares about impactful communication of advances and solutions. So let's go ahead and get started. I'm going to kick it off with Dr. Marks. So you've been very vocal about your vision for the future of the regulatory process for gene and cell-based therapies and how accelerated approval provides a pretty awesome mechanism, but it also presents a series of opportunities for the generation of novel evidence and solutions. Can you comment on that?
Yeah, so, okay, so for cell and gene therapy in particular gene therapy, I think this is an area where it's kind of one of these marriages made in heaven, right? It's like chocolate and peanut butter. Accelerated approval, gene therapy, biomarkers, and go very nicely with gene therapy. Why? Because very often when we're dealing with gene therapies, we actually, just the fact that we have a gene therapy means we understand something fundamental about the disease we're trying to address. We understand the gene that's causing it. We may not understand all of the aspects of the biology of that gene, but you know, there are three categories three major categories that we have. There's the category where the gene therapy product is the concentration of that product relates directly to the clinical outcome. Think hemophilia, right? So if you can measure the gene output of the gene therapy as a biomarker, you have something that's reasonably likely to predict how someone's going to do clinically. I'm a clinical, was a clinical hematologist, oncologist for many years. you know that once you have factor levels above a certain level, people just don't bleed. So they're easy, right? So nice, easy biomarker. There's the issue where sometimes you can't measure the actual biomarker gene therapy product but you can measure something in the pathway upstream or downstream of that biomarker. Perhaps the gene therapy is a structural protein or an enzyme that you can't measure. But there again, if you can, as long as you have something you can measure, you've got a hook. Now, there's the third case where you're unlucky and you don't have something to measure. That sometimes happens in the CNS space where you have a tissue that's inaccessible, that you can't get to the gene therapy product easily. That gets you more back towards more traditional development pathways, or you have to be novel in how you do things. And there are examples of that. For instance, otic gene therapy, where you can look at auditory evoked potentials, where sure enough, you don't need to measure the gene therapy product because, again, there's a very good correlation with expression and reasonably likely to predict. So I think we have a situation with gene therapy that links us nicely to the ability to use biomarkers and sometimes intermediate clinical endpoints to get there faster, to get these products to people more efficiently. And in a lot of these rare diseases, the idea that you're going to get it's absurd to think that we're going to do randomized clinical trials on populations of 30 people or 40 people. It's also very challenging, even if you had slightly or somewhat larger patient populations, you have this very strong urge to try to do something for children who in some cases are otherwise having severe consequences. And they have a third point, though, which is that for gene therapies, the other reason why it's very amenable here is that single arm trials are possible because if your gene therapy doesn't make a significant difference, like 25, 50% difference in that biomarker, you probably shouldn't be developing that gene therapy. In other words, if you can't see, you shouldn't have to get out the microscope to look at the differences between the curves. I know that was what, when I was a medical student with all the TIMI trials, it was always so exciting to see the next TIMI trial where you'd look in the New England Journal and there would be about this much space. If you can't see any space, you're about right between things. Here with gene therapy, we're looking at something different. So it's a combination of a variety of things that make gene therapies fit nicely towards the accelerated approval pathway with biomarkers.
Yeah, that's a fantastic point, because that opens up the connecting the dots opportunity with understanding what the solutions need to look like, AKA what's the context of use statement for each one of the solutions. So I'm going to go to Jeff because you live that life every single day in your leadership of the Office of Drug Evaluation Science. Can you comment on the importance that you place on defining but also evolving the context of use statement in the process of the review and as evidence is being generated? Yeah, you have a
Okay.
okay.
First of all, it's very nice to be here. My name's Jeffrey Siegel. I'm Office Director for the Office of Drug Evaluation Sciences in the Center for Drug Evaluation and Research at FDA. Our office is responsible for qualifying biomarkers for a variety of different uses, also qualifying clinical outcome assessments and other drug development tools that can help in the drug development process. When we think about qualifying a biomarker for a particular use, we think about what type of biomarker it is and how it's going to be used in the drug development process. So the type of biomarker would be, for example, a diagnostic biomarker, such as alpha-synuclein seed amplification assay, which is an important new tool for identifying people with Parkinson's and even people with early prodromal Parkinson's. That would be an example of a diagnostic biomarker. And then there are prognostic biomarkers, which identify the patients who are most likely to progress with the disease. These are very important for enriching clinical trials in patients most likely to have an outcome so that you can get an assessment of efficacy with a smaller, potentially shorter trial. The last category of biomarkers that's relevant here are surrogate endpoint biomarkers. These are ones that are used as a basis of drug approval. Think low density lipoprotein for hypolipoproteinemia. That would be an example of a validated biomarker, or hemoglobin A1C. And then there are reasonably likely surrogate endpoints, which are used as biomarkers for accelerated approval for serious or life-threatening conditions. And in that situation, if the drug is approved on that basis, you also need to do a confirmatory trial to show that it works. I guess the one other thing that I would, well, okay. So in terms of context of use, we think very carefully about what type of biomarker it is and how it's going to be used in drug development programs. And being very specific about what the context of use is very important. And it does sometimes evolve during the course of a program evaluating a biomarker. For example, a biomarker might begin as a prognostic biomarker, but then you might find that it changes with treatment, and that change anticipates change in the clinical outcome. And then it could potentially be used as a reasonably likely surrogate. So that would be an example of the way a biomarker's context of use may change as new evidence gets accumulated.
Yeah, fantastic point, Jeff, because you just illustrated how the evidence evolves. So that's an illustration of the learn and confirm paradigm. But a lot of times you're going to need to pay attention to longitudinal analyses of disease dynamics and biomarker dynamics, a lot of times together. So yeah, you live in a world in which that happens day in and day out. And some of those tools can also become clinical trial simulation platforms, for example. And you represent the office that leads a specific pathway for the review of those types of tools. Can you comment on your experience of how it's also really important to understand the context of use for those quantitative solutions?
Exactly. So first, I'd like to thank you the insight question and invite me for this wonderful workshop. Something about myself, I noticed a typo for my credit. So I'm not from UCSF, so I'm a associate director in the Division of Pharmacometrics, Office of Clinical Pharmacology, Office of Translational Sciences, CEDA. So there's a typo there. And also, so we already have great discussion about the biomarker, COA, or those innovative methodology for digital health technology and maybe AI machine learning. So for my background, I'm a pharmacometrician here. So we are mainly dealing with, like you mentioned, those dynamic models via the fit for purpose pathways. So fit for purpose pathway initiative is a pathway for the regulatory agency to accept those dynamic models, like classes is those disease or drug or trial models, which may change when you have like additional data available or new information for those important covariates, and also like knowledge regarding the target mechanism and even the drug and the trial conductions. So I think there's a lot of opportunity for the collaboration here. We can see like a multiple stages from this tool development. At the beginning, we need to work with like a multidisciplinary, especially on medicals, to define, to specify the question of interest, then clearly define the context of use. So we need to make sure those questions is like to address the clinically critical area that have strong unmet medical needs. Then this context of use should be very clear, defined, and then specify the role of these tools to ensure it can at least partially answer this question of interest.
Yeah, fantastic, because that leads me to Cecile. You care about the global aspect of drug development, but also the global regulatory pathways. So can you comment on how you see the alignment between multiple agencies regarding the notion of truly understanding the context of use statement and the evidence behind it?
Yes, so maybe I can say a few words about my background because I might have a bias in my background. So I'm working for the Critical Path Institute for 3 years now, and I'm the director for the European office. But prior to that, I was, amongst other things, for 11 years at the European Medicine Agency, where I worked a lot on pediatrics and in ICH with Lynn Yao and Solange on the topic of extrapolation. And there are lots of things we have understand in the pediatric population or in rare disease where you truly need to facilitate this global alignment or collaboration in order to facilitate access to innovative therapies. And I think that, okay, each agencies have their own pathways and practices, and we need to respect that. And that's linked to the regional culture. And but we can work around that. And I think in order to, what would be important, as we have seen in the review of some drug development tools that are developed in the US, because the data that are the source data behind the development and the validation of these tools are also linked to the different regions. So if you have a DDT that is in an area, a model for a rare disease that is being developed in the US, you go through all the regulatory pathway in one region, you get eventually validation, and then you engage with the other regions. But meanwhile, you don't necessarily have the data from the other regions. And this can create a lot of delays and also therefore have different perception or interpretation behind the use of these tools. So it would be important in the early days when we plan to develop a tool that we know is going to be in an area where there is a global impact, whether pediatric, rare disease, but also Alzheimer, Parkinson are these areas where we truly need to collaborate and sometimes have one model like it was discussed yesterday for a given community. It allows to align the purpose of the tool and a well-defined context of use from the early days, and then gather the questions from the two regions in a coordinated manner that allow for the two agencies to reach a compatible opinion and have a tool that is readily usable by both regions, and even beyond. When the two regions are aligned, we know that it's of benefit to all the other regions. So I think that is something that is important to, and why it's important to develop these tools in the context of public-private partnership, because there truly are so many parameters to be considered, because we can, I mean, maybe I think, yes, it's easy to develop a tool, but a tool that is of use, it's another story. So, and that's where the efforts should be invested to make sure that this tool then can truly have an impact and be of use by both regions and communities.
Absolutely, and I'm gonna push you even further, because it has been our experience that when you peel all the layers of the onion, I have seen nothing but agreement on the science and the clinical relevance when we are discussing context of use, data, and evidence between FDA, EMA, and PMDA. That said, the agencies are bound by their jurisdictions and their laws. And that's something that could be pushed by lobbyists and legislators, but that's a different discussion. I want to make sure that then I turn to Carla, because you and I were having a conversation about something that our founder taught me like on the second day on the job. Solutions that are not adopted are not solutions. So people need to know about the solutions. And you're in an organization that really cares about the right dissemination of the right information so that people know that these solutions exist. Connects the dots to what Rob Califf was mentioning day one. So can you comment on your experience on serving as that communicator of the existence of these solutions?
Sure, I'm happy to. So as Klaus said, I'm Carla Rodriguez-Watson. I direct a research group at the Reagan-Udall Foundation for the FDA. So if you've never heard those words strung together, we are the FDA's foundation. We're a completely independent nonprofit, but uniquely created by Congress to help advance the mission of the FDA. So I see the value in public-private partnerships in helping to both develop and disseminate these tools. In development, public-private partnerships really have an opportunity as not just being a single host, but dual host partnership in inviting a really diverse group of stakeholders to get to the table. The data generators, which are the health systems themselves, the patients, importantly, the clinicians, to be able to share their perspective in a neutral space where they can feel free and not feel bound by any kind of formality to say or not say certain things. Secondly, in the development, as this convener, these folks can say things again and we can help the agency hear things that they need to hear, but not necessarily are able to ask themselves. And having this diversity really helps come to a problem from all different angles. In terms of dissemination, a lot of these tools, including frameworks, come in the form of a framework, right? That isn't necessarily a recipe or a way to operationalize. So again, in being able to bring together diverse stakeholders, we can work through that framework through either case studies or a specific aspect of it to really get at how do we operationalize this and apply it and make it real and pressure test if there are additional questions that need some clarification.
Awesome, because that brings me back to Peter. You and I were having a conversation about the 4 pillars of regulatory decision making. The clinical context, the full evidence, the remaining uncertainty, and the regulatory pathways. So 2-part question. Number 1, how do you see the role of real-world data and real-world evidence with the future of drug development, what CBER is going to see in those pillars? And then probably the other 2 pillars that we need to consider for gene and cell-based therapies, which is manufacturing and reimbursement. That's the second part of the question.
Yeah, okay, so 2 meaty questions. Real world evidence is something that we've been using at the center already for years. Obviously, it's what we use in pharmacovigilance for vaccines and has gotten us much of the safety data. For instance, for the COVID-19 vaccines were gathered with real world evidence. But before that, before the pandemic came about, the COVID-19 pandemic, we had done studies actually showing that you can use large databases for vaccine effectiveness studies when you have an outcome that's very apparent. An example was looking at high dose versus standard dose influenza vaccine in the elderly or older individuals, people 65 and up. There you have the CMS database, very nice database to be able to use. You can compare those who got the standard dose vaccine versus those who got high dose vaccine and look at hospitalizations. And it turned out that real world evidence study actually mirrored what was done by the manufacturer of a particular vaccine within about 2% in terms of the outcomes, although they were actually not able to look at hospitalization. They had to look at influenza-like illness. So actually you had a better hard endpoint from the real world evidence. Now, the problem is we don't have 2 things in many cases. First of all, we don't have large databases for many of the things we're looking to study. And even if we did, the data quality of many of our databases today is simply not that of what we would need to do for real world evidence. so you can use them in the oncology setting. It works because death, it's pretty easy to figure out if somebody is alive or dead. Progression-free survival even is something that you can, is possible. Although even that gets to be challenging for medical records if you have to review scans. But more fine outcomes measures, it's very challenging. And it would be really nice to be able to use real world evidence in the evidence generation phase of things up front, but it's just a challenge. In oncology, it's being used for second, third, fourth indications because you can do it. I think the soonest place I can see it applied in the gene therapy space is going to be in moving from accelerated approvals to traditional approvals, where there it may be possible for real world evidence to be collected to help us. There you can use hard endpoints that should be measurable. It's a relatively small number of patients. You'll probably be able to get consent to look longitudinally along their medical records, and people are working with blockchain keys, et cetera, to help you be able to move, to watch the people as they move across the country or with different health plans. But those where I see kind of it would be really exciting. It would be wonderful if suddenly we could have like magic wand and poof, the United States had a medical record system like Denmark, where on any given day, if they want to know how many people had a sore right toe, they can, push a button and they can find out. We can't do that here. It's probably a ways in coming. Now, your second question was this manufacturing issue and reimbursement. And I think increasingly, I've been thinking about gene therapies in reimbursement, not our issue at FDA. On the other hand, the idea of having an ecosystem that's there where people can't actually get treated because we can't afford the products is a problem. And so getting to the right place probably is a combination of figuring out how we manufacture more efficiently and effectively in this cell and gene therapy space, which has been a huge problem. And it remains a problem because these are basically niche products, small numbers of doses that are developed. And the model is very jarring to those in this space. Why? Because think about a gene therapy. If you have a given genetic disease, you develop a gene therapy for it. And although some diseases like hemophilia, there is a natural spontaneous mutation rate, so you see new people coming up. Many of these, you develop a gene therapy, there's kind of a pent-up demand, you meet that demand, and then what happens? As opposed to the normal net present value, normally net present, for those of you, here's the 1-minute business school. Normally, pharmaceutical companies, they do net present value calculations, and the net present value is based on out years, right? A lot of the value is based on out years, where before the patent expires, the last year or two, boy, that's where here the problem is, what happens is a lot of your income comes upfront and then it tails off, potentially, if things were really working well in gene therapy, which I'm not saying they are just yet in terms of being able to deliver. But this is a problem we have to address. And some of it is by being able to figure out how to manufacture more efficiently and effectively. Probably that has to do with getting towards automated manufacturing or more centralized manufacturing. And then the reimbursement is probably going to have to be baked into this. In some cases for gene therapies, as we start to think about low and middle income countries, my guess is, again, not our problem at FDA, so I'm not trying to go into CMS's domain, but probably people will have to be thinking about the cost as they develop a gene therapy, because to say you're going to develop a gene therapy that you're going to deploy on the African continent that costs $2.5 million per dose means it's not realistic. You're not going to treat any significant number of people. On the other hand, if you really do want to do that, maybe it's time to start doing the calculation of what you can afford and work backwards and figure out what you have to do for manufacturing efficiencies. But it's a real challenge because even here in a high-income country, gene therapy has presented a real challenge because many of these gene therapies are not forever cures. They are 10-year, 15-year treatments. And so, and we don't know how long they're going to last. And so insurers who are going to put up a lot of, now somewhere between $1 and $4 million upfront don't know what they're going to get in return. And so it's been a real challenge here. And the other problem is that we have is that in the United States, about every 3 years, people switch insurance. So if I pay as an insurer $2 million for somebody's gene therapy, well, they go someplace else. Hopefully everyone's getting that same, it's equally distributed, but not necessarily so. So it's a very challenging area. And I think, although as a regulator, we are tasked with, I just was looking at the Biologics Control Act of 1902 this morning, what I do in my spare time. It's the safety, purity, and potency of the products that we regulate, and therefore also their effectiveness. I think if we don't think about it more in terms of the totality of everything from manufacturer on out to how it's going to get reimbursed, and then ultimately the long-term effectiveness, I think we're not doing our public health a full service.
And while we fix the mic issue, thank you for that, Peter. That really illustrates a set of opportunities that we have for novel evidence generation. Yeah, I think it ran out of battery.
Battery. It says it wants a battery. That's bad.
Or called Batman. So it illustrates a really important point about the opportunities that lie ahead. So another thing that our founder used to say, there aren't challenges, there's only opportunities. And so this is the kind of opportunities that lie in the future, and we're not going to stay stagnant, and we're going to seize those opportunities again, leveraging the public-private partnership model for the benefit of the people that need that kind of new evidence. I'm going to turn to Jeff now, because with the illustration that you gave about context of use and having that clarity, not just for biomarkers, but also for COAs, that doesn't necessarily imply that it's just about the clinical space of drug development. There's a lot of opportunity also in the preclinical and translational, and you've seen some of those case studies on both ends. Can you comment on the case studies you've seen and some of the more successful ones?
Sure. So the qualification program accepts submissions from people developing drug development tools for anything that can potentially help drug development. When we receive these requests, we look carefully at the context of use and make sure that it's something that fits in the regulatory purview of the agency. When it comes to the preclinical space, many of the things that come in have to do with drug discovery and screening compounds that pharmaceutical companies are thinking of taking forward. We don't generally get involved in that area. On the other hand, putting together the safety database to show that a product is going to be safe when introduced into humans is very, very important. And one area that's beginning to emerge is the area of microphysiologic systems. So these are systems in vitro which look at cells and tissues from organs that are under study, and sometimes systems of organs, not in 2D plates, but in a 3D form, which more closely mimics the real life situation for the organ. So these can consist of spheroids, sometimes tissue on a chip, and these can be used to evaluate pharmacokinetic properties, potentially drug-drug interactions, and potential toxicities of drugs on a particular organ. I'll give you one example of a program that we've recently accepted into the iSTAN program. So this is a product from emulate that is a liver organ on a chip, and it includes the relevant tissue, cell types from the liver on a chip. And when you expose it to compounds, it gives a readout of potential toxicity of that compound on the human liver. So we recently accepted this into the ISTN program for a particular context of use. So in some situations, there are drugs that have been dropped because they had a liver injury signal when introduced into humans. Now, these are drugs that presumably didn't show any toxicity in animal studies, so they were introduced into humans, but then surprisingly, they showed a liver injury signal in humans. And then other compounds come out that are similar in chemical composition, but different in a way that the developer hopes will get around the liver injury signal. And what Emulate is trying to do, and the reason we accepted into the ISTN program, is that it has evidence that this liver on a chip system can differentiate new compounds that are like compounds that were liver toxic that aren't liver toxic. So we hope as it goes through the qualification program and provides evidence in support of this, that this can be one tool to assess whether compounds are liver toxic in humans. So this would be one example. We have other examples of drug development tools in the ISTN program and the biomarker qualification program. One is this, and a lot of these have to do with the FDA mandate to it's called the 3 Rs, to reduce, refine, and replace use of animals in non-clinical testing of drugs. So we have another program that's been accepted into the ISTN program to look at the possibility of toxicity of products that are going to be introduced into the CNS, into the CSF, the cerebrospinal fluid, and leachables from the tubing, from the needles, and so on, can sometimes provoke inflammation or other forms of toxicity in the surrounding neural tissue. Usually, these leachables are tested in animal systems, and the system that was accepted into the ISTN program tests these compounds in a series of in vitro systems to see if the leachables are going to be toxic in humans. So this is another example of a drug development tool that will give better evidence of safety of compounds that are going to be introduced into humans that also accomplishes the goals of the 3 Rs. And there's another system that was recently introduced into the biomarker qualification program that looks at MRI of animals in the in-life setting for non-clinical testing to see if these compounds are potentially toxic to the CNS, to the CNS. And this can serve as a way of screening compounds for ones that might be toxic, and also figuring out where the toxicity might be so that you can do more refined testing on histopathology, for example, to see if there is indeed toxicity in those areas. So these are a couple of examples of how safety testing can be accomplished as part of the qualification program. And there are others that relate to looking at in-human testing, but did you want me to go into those as well, or?
Sure, yes.
Okay, you had mentioned the non-clinical, but there's a tool looking at GLDH to look at liver toxicity in situations where the people being tested may have muscle enzyme elevations that can cause confusion between the tests that are looked at for liver toxicity. And this would be a test that's more specific for liver and gets around the problem of non-specificity of the other usual enzymes that are tested where muscle injury can cause elevation of those. So this would be another example of a drug development tool that can give more precise information about potential toxicities of drugs.
Yeah, and that's a fantastic point, because you just saw Peter and Jeff teeing up a couple of sessions later today. Number one, a real world data session in which the neonatal field is the one teaching everybody else how to do it with real world data. And you're going to see the case studies and the case examples of 300,000 individual patient records from neonatal intensive care units globally. US, Japan, Europe, all contributing those data for the advancement of solutions that matter in drug development for that space. And then Jeff teed up the session that is coming up on novel and advanced methods to continue to transform translational sciences to make it truly, truly translational. And he illustrated a point. I'm going to steal a phrase from my colleague, Nick King, who's going to be leading that session. The safety biomarker of today could be your efficacy biomarker of tomorrow. And that's an example of the linkage between GLDH for safety, but also a component of efficacy in Duchenne muscular dystrophy. So those were beautifully teed up components for the sessions later. So thanks for connecting the dots. Now, Jian, so we always talk about the notion of simulations to inform trial design and to optimize trial conduct. But there are actually examples in very specific context of use situations in which the simulations, especially if they have relevant mechanistic components, can actually replace the execution of actual clinical trials. I'm talking specifically about PBPK in the context of special population studies, and how a lot of those can be run purely in silico today, and how those have contributed to labeling refinements for drugs in more than one occasion. Can you comment on that as well?
Yeah, sure. Yeah, if you want to use simulation to replace actual clinical trial, the bar is very high. So we need to make sure the model actually is reliable and they have prediction power. Also, so most of the tool we are talking about haven't to reach that stages, but you mentioned in several scenarios, we can do the extrapolation, maybe especially for pediatric extrapolation sometimes, or the new formulation, or changing dosing frequency, all those things. Maybe we can do this based on simulation, but still, if it's like an intraoperation, maybe the regulatory is feel more comfortable. If it's extraoperation outside the range, we still, especially clinical, may still want safety experience to ensure we don't have additional safety signals. Yeah, we have several. One without a clinical trial, we did a simulation, especially like the dosing frequency change. Maybe you can change it for PD-1, PD-L1 drug. We have that guidance. You can change it dosing frequency based on the PK match, and change it from like every 2 weeks to every 4 weeks, or those things. PK, yes, if you already have those mechanistic understanding of the drug interaction, then you can do some extrapolation if you already have data based on the strong inhibitor or inducer. Then in the middle moderate one, maybe we feel comfortable to do the simulation, make a decision based on. So these are all like risk-based evaluation. We need to evaluate the model risk according to the model contribution, whether it's solely based on the simulation or you have additional data to support it, then we can use totality of evidence. And also the decision consequence, what is the consequence if you made a wrong decision? So if it's based on those things, we will evaluate the risk and decide our strategies.
Yeah, fantastic point about the full package of evidence. And you touched on extrapolation. And so, Cecile, you lived in that world for quite some time and you saw both sides of the ocean. And then from a global perspective, can you comment on your experience driving that?
Yes, and I think that so all the points you mentioned are extremely important because when you discuss the impact of the model on the regulatory decision making, particularly models that are developed to replace data, I think that a bit going back to what was said on the first day, like I think it's important to go stepwise and make things a bit more palatable for the systems and minimize risk and uncertainties. And that's a bit also linked to the process we were discussing on how to develop a drug development tool and have the context of use that is well defined from the beginning and have sustainability aspects also from the beginning clearly present in public-private partnership to allow to have this context of use life cycle that may be allowed to start with a context of use that is more realistic than replacing the generation of data and can inform the regulatory decision making in a way that data are not entirely replaced, but that can help to make informed decision on the prediction and better understand what are the uncertainties, how can they be addressed in the post-marketing as well, and have a clear long-term strategy that will inform the evolution of the context of use and minimize the uncertainties that are linked to the regulatory decision making. And I think particularly in pediatrics and rare disease, that's something that the extrapolation framework can help. And also maybe I will take 1 second because we said today pediatric rare disease. I want to congratulate Lynn because the ICH guideline on extrapolation have been adopted recently, and it's really a big win because extrapolation, it was the same when the PDCO products were being reviewed and the clinical context was there and there were confidence from the clinician, okay, we can extrapolate, and in the pediatric plan, the extrapolation concept was not clearly laid out, and it has clearly a major impact since it was avoiding the generation of data. When it was reaching the CHMP, where the products were licensed and there was no clear understanding behind the rationale, the lack of communication with the years, you have between the design of a pediatric plan and the licensing created some challenges. That's what the extrapolation framework helps to have truly a life cycle of your plan. And I think it's the same with the context of use, like start to identify where are the gaps, what do you need to address, when do you need to have trials being designed in a way that will generate the right data to minimize the uncertainties, and go stepwise, and then you reassess. And I see similarities in the methodology behind the extrapolation framework and how we develop model in order to facilitate the regulatory validation of these tools, not necessarily linked to the to an opinion, which takes time. But yeah, I don't know if that answers your question.
Perfect point, because that'll bring me nicely to Carla and how you see a future for Reagan-Udall in the next 3 to 5 years in owning a lot of the right dissemination at the right dose for multiple audiences for all these concepts that sound really complex, but that can be and should be readily explained to multiple types of audiences. Can you help us with your vision for that?
Yeah, I think Dr. Marks, you touched on it when you're talking about cell and gene therapies and real world data. I think the use case for us to really think through how public-private partnerships can help build kind of a learning hub for evidence generation is around artificial intelligence. I mean, we're thinking about deploying this for cell and gene therapies to identify valid and invalid targets. And so with that, creating, taking a piece from what clinical care has used in the learning health system to develop a robust evidence generation system that includes the data generators, meaning the health systems themselves, the patients, the clinicians, the epidemiologists and statisticians, as well as the life science industry. I had a whole list here because I want to make sure I didn't miss anybody. Having the regulators, informaticists, the lawyers, very important if we're talking about getting information exchanged across places so that we can simulate Denmark's like health records, since we don't have that in the US, an ethicist, because we are talking about patients' data and really having patients weigh in on what are important outcomes and endpoints for these trials that will be reflected in these biomarkers. So all that working together, I think is something that Critical Path, Reagan-Udall Foundation, our folks at FNIH, we all need to be coming at this elephant from different parts and helping to elevate the great tools, frameworks, thinking that's coming out of our regulators.
Yes, and through the public-private partnership approach, have consistency in the message of all those parts of the equation.
Right. Because there's a lot of energy around this right now, and from all parts of the U.S. federal government. There's ONC, the Office of the National Coordinator. Dr. Marks talked about this need for us to really talk to the regulator. Again, we're not your job, but there is more an effort to really bring clinical care and clinical trials closer together. And that also means having the data be relevant, reliable, and transparent and interoperable. So there's a lot of incentive frameworks, guidance happening right now.
Fantastic point. So, Peter, where do you see the biggest potential set of contributions from public-private partnerships to help CBER succeed in your vision for the next few years?
Okay, now we have a working mic. So I think there are probably two places. One, you guys may care less about, the other you probably care more about. The one where I think we'll see hopefully some public-private partnerships is in this area of gene therapy manufacturing, because there are some very specific things that need to happen. One of the issues is working out how to actually do the technology transfer to scale up from academic small batches to large batches. Mm-hmm. For some reason, it's been very hard to get people interested in doing that. It shouldn't be so surprising. Academics aren't going to get papers out of it that really are going to get published in top tier journals, and industry makes a lot of money out of doing it. It's like if they do it fresh each time. I mean, with all due respect, they make more money that way, and it's a good business model, but it's not good for the ecosystem. And so I think we're going to have to figure out a way that industry can still make reasonable money, but we do it more efficiently. If you do something more often that costs less, you still make a reasonable profit. I think that was another thing they taught me in Business School 101, how to be I didn't go to business school, but in Business School 101 class. The other piece, though, that really is for us is that I think increasingly as we have gene therapies proliferate, we're going to have two sets of products. We're going to have people who are going to have been treated with a gene therapy that will have been investigational, that will never have made it across the finish line to an approved product. Those people will need long-term follow-up, and it will be important to follow them up to understand the adverse event profiles of the particular gene therapy type that they received, whether it be a viral vector or non-viral vector. And then there will be licensed gene therapies that will want to understand how people are doing over the course of time, for two reasons. A, we want to see the effectiveness for that individual disease. But when you step back, a given type of gene therapy, for instance, an adeno-associated viral vector 9 vector gene therapy, the backbone, it may be that there are things that will be relevant to the AAV9 platform or just to AAV in general that we may learn if we have this longer term follow-up. So I think the partnerships that we'll see are trying to get those data to be able to collect them on those who are being treated. And I think it's going to be a tall order because there are a lot of different companies, a lot of different investigators out there. So I think, but we really do need to work to do that, because otherwise we're not going to develop the knowledge that we need to understand whether there are some benefits that we're not seeing or some risks that we're not seeing in the future.
Fantastic points, and I will thank you for teeing up the other session of the day about cell and gene-based therapies. But what we've observed is that this classic divorce between development and manufacturing can be divorced in cell and gene-based therapy. So, and there's a lot of potential solutions and tools that can help both. And so that's going to be part of that conversation also, to help support that vision. Now, Jeff, we've talked about a lot about the evolution of qualification. Where do you see public-private partnerships helping your vision for the future of the reuse of these drug development tools?
I see two main areas where public-private partnerships can aid in validation of drug development tools. One is to pull the evidence together to qualify biomarkers for use in drug development. And this would be as diagnostic biomarkers, prognostic biomarkers, validated surrogate endpoints, and reasonably likely surrogate endpoints. They're one of the key pieces that a public-private partnership can bring together, is to bring the resources to bear to pull the data sets together, to analyze the data of the biomarker, connecting it to potential clinical outcomes, carrying out the modeling and data analytics that are required, which can be quite resource intensive. Another important role is, of course, to bring the different stakeholders together, including industry, academia, regulators, and bringing in the patient voice as well. So validating biomarkers is one key area for public-private partnerships. I think the other area, which I see as a really important area for the future, is digital health technologies. Digital health technologies just have a tremendous potential to collect important information for patients and for clinicians about the effect of new therapies on important diseases. These include wearables, where you can measure important clinical outcomes in the patient's real world, and not just in the laboratory. And you can do it continuously over time, so you can see if the effects are sustained, and quantitate the changes. This is very helpful in many cases where ordinarily you have to infer the effect of the drug on the particular condition based on patient reports, which can be subjective, and having the objective data to back that up can be really, really important. But developing digital health technologies as a tool that can be applied in drug development programs is complicated. And this is a really important role that public-private partnerships can serve. So if you're developing a drug development tool to measure steps in, for example, heart failure with preserved ejection fraction, well, how do you know if a 30% increase in the number of steps someone takes is really what's clinically meaningful to the patient? It may not be. What may be important to the patient is their ability to go shopping, their ability to walk to the store, their ability to do gardening, vacuuming, whatever is important to them. So collecting the information about what's most important to patients and how that can be measured by the digital health technology requires a number of different stakeholders, and pulling the data sets together to validate them. I think that's a really important role that public-private partnerships can serve. And then bringing in regulators to be part of the process, and to be able to talk about some of the strains and some of the potential limitations of the process that's being taken, can make sure that the efforts go on track so that the resulting tool can be applied effectively in drug development programs.
Fantastic. Gian, connecting the dots to what Patrizia Cavazzoni was sharing with us on Monday regarding the efforts, the Fit for Purpose initiative, the MIDD program, and as of more recently, the Quantitative Medicine Center of Excellence. Where do you see the role of public-private partnerships in this future of integrating quantitation into drug development in a really meaningful way?
Yeah, actually, I don't have much to add. Dr. Siegel already gave a lot great perspective. Also, for the last 2 days, we already have very intensive discussion on those things. Although may not be specific to quantitative part, but I guess we see the trend, like we see we have increased availability of data come from different sources. We see this novel therapies like gene therapy or cell therapy, and also the RNA, this oligonucleotide, and also this bispecific ADC, all those coming together. Also, we see this novel innovative endpoints, COA, and also biomarkers, and also this new technology, DHT, and AI machine learning. So I see there's a great opportunity for collaboration involve all stakeholders, even for quantitative approaches. And basically, like Jeff said, many folks, maybe I see two major areas. That is for resource sharing. So this is, besides the data sharing, we also have knowledge sharing. So there's a lot of expertise, especially for rare disease. Now we have more than 10,000 rare disease. Most of them involve pediatric, and we don't know actually the disease progression or the mechanism behind the physiological mechanism behind it. So I guess we need multiple stakeholders work together to understand the target, the disease, all those things. Then we can develop like QSP model, other things, maybe to characterize the disease progression. And also we need to understand the drug action and the trial conduct. So all those things. And another part, I think we can work together for education to raise the expertise. So now we are lacking a lot of experts in this field. I guess we can work together to increase our power of those knowledges.
Fantastic. And I'd be remiss not to mention the fact that on the FDA's YouTube channel, there is a pretty amazing MIDD training course that we developed together with the agency and experts in the field for scientists of any background. This is not just for us nerds that go to ACOP in November, even though we are going to nerd out and have a lot of fun there. This is for everyone. This is for everyone. So make sure that you connect with Jan or with any one of us, and we'd be happy to share the information for that training course. My colleague from CPATH, you get the last word. Where do you see the future globally of public-private partnerships?
So I have nothing to add to what was said on the areas and the needs. What I see between the different type of public-private partnership in Europe and in the US, what really is being seen as a success in the US from Europe is the collaboration between FDA and CPAS over the years, because it has brought the sustainability component in this public-private partnership. And all the challenges we know that are related to public-private partnership are operational around data collaboration, all the expertise you need around data collaboration, the infrastructure you need to have this database coming together, facilitating interoperability. So I think if we manage to keep that sustainable infrastructure, this one-stop shop for regulatory science, data collaboration, biomarker, all these aspects that are being dealt at CPAS, in a sustainable manner with now the European office facilitating the global collaboration. I think any challenges can be taken upon, and there will always be new challenges and new needs. And I think what is important is to address them in a way that is cost efficient and agile. So, that's where I.
Beautifully
think
stated. With a few minutes to spare to get the next panel up and running, please help me thank the panelists for a fantastic conversation. Thanks, everyone.